Okay. So first of all, I would like to thank the organizers really for giving me the opportunity to share with you our results. And what I would like to do today is to discuss with you a dilemma that we are addressing and describe the approaches we are taking to address this dilemma. And most importantly, what I would like to do is really to point to where I hope to get some input and maybe establish new collaborations on this work. So basically, I'm going to speak about mast cell activation, more specifically about signal transduction-directed drugs, and even more specifically about cell-permeable peptides as potential anti-allergic and inflammatory drugs. So background is really simple because I think even in a heterogeneous audience, most likely each one of you has experienced an allergic response. And the bad news are that whoever did not is likely to experience one in the future. Because allergic diseases, especially the reoccurring chronic allergic diseases, have reached really an epidemic, have reached epidemic proportions, and according to the internet, they affect currently about 30% of the world population. And this number is increasing, and there is a hypothesis that the cleaner we get and the more we keep clean and our children clean, then we are more likely to be exposed to allergic diseases in the future. So 30% of the world population is affected by allergic diseases, and it has become the fifth leading chronic disease in the world. Yet, in terms of treatment of allergic diseases, and as we shall see, when we are speaking about allergy, we are speaking about a family of diseases which includes skin diseases, skin allergy, food allergy, rhinitis, which is nasal allergy, namely respiratory allergy. All of them, in fact, are the same disease and share the same molecular mechanism, which is important to appreciate if we are trying to think about ways to treat these allergic diseases. And yet, as you may know, in fact, allergy treatment, there isn't really a cure for this family of diseases, and what we are really doing is to deal with the symptoms of the disease and not really with the disease itself, and this is because of the complexity of this disease. Now, the major players of these diseases who mediate all these allergic disorders are cells which are called mast cells. And it's important to appreciate that those are the same cells which mediate all the allergic diseases which I just mentioned, because these are cells which are derived from the bone marrow. They invade tissues where they differentiate, mature, and become mast cells. And so we find them distributed along our body, especially on those surfaces which are exposed to the external environment. So we have mast cells on our skin, we have them along our digestive tract, we have them in the respiratory system, and more. So it is the same, those are the same cells which are mediating the allergic responses, and therefore, if we are speaking about skin allergy, we mean that mast cells in the skin are the ones that are mediating the disease. If we are speaking about food allergy, it will be the mast cells in our intestine that are involved. So these are the same cells and the same molecular mechanisms which bring about the allergic disease. So how is it really mediated? How do mast cells, the principal players in the allergic disorders, how do they really mediate an allergic response? So as you can see here schematically, the mast cell is a cell that has many vesicles inside. Those are referred to as secretory granules, and these vesicles are really filled with biologically active materials. So they have many substances which are pre-stored in these vesicles. Most known one is histamine, but it's not only histamine. There are many substances, all of them of biological activity that can affect blood vessels, muscle contractions, attract other cells from inflammatory cells and bring them along, all these materials are pre-stored in these vesicles, and they are referred to as class 1 mediators. Now, when this cell encounters a trigger, and we'll discuss in a moment what we mean by a trigger, when this cell encounters an external trigger, it becomes activated, meaning that it then undergoes a process where it releases the contents of all these secretory vesicles. So from this stage, it goes to this one, secreting all these materials which were previously stored in the cell and now are extracellular, ready to affect all the other target organs or cells, bringing about what we call the early immediate response. So within minutes, since the cell has encountered an external trigger, it will release the contents of its granules, and because these are biologically active materials that will affect other cells, we get an inflammatory response, which we call the early immediate response or the allergic reaction. Now, if this happens within minutes, the cell continues to respond to this similar trigger, and within 3 to 4 hours, it produces new materials which were not there before, which are also of an inflammatory nature, prostaglandins and leukotrienes. They will be released as well, the class 2, and again will contribute and amplify the inflammatory reaction. And within 6 to 24 hours, even more biologically active materials will be produced. This will already involve, at this stage, gene transcription, formation of new messenger RNAs, new proteins, and secretion of new molecules, cytokines, chemokines, growth factor, molecules that will be released from this cell and again will interact with the environment and will amplify the response, leading to what we call the late inflammatory response. So an allergic reaction has two phases, an early one, which is immediate within minutes, and then a later one, which is an inflammatory one, involves the recruitment of more inflammatory cells, giving rise to what we call an allergic response. Now, how about activation? What is, in fact, this green? What is the trigger for this reaction for the mast cell to begin releasing its contents and to produce also the class 2 and class 3 mediators? So already from this cartoon, you can see why I said that this reaction is really a complex one, because there isn't one single trigger. There are many, many ways to activate a mast cell, and therefore many, many ways to trigger an inflammatory and allergic reaction. The most known one is the immunological response shown here, where an allergen, which could be a completely innocent material, for some reason in the allergic person, leads to an immunological response where this individual will produce antibodies against this allergen. So usually we are supposed to produce antibodies against something that could harm us, like bacteria or viruses. But here it's an allergen, which could be food or any other material, will trigger this immunological reaction and produce specific antibodies, not the regular IgG, but antibodies which belong to a different class. They are called IgE, which are produced and are specific for the allergen. They bind to specific receptors which are present on the mast cell. And then when such cells encounter again an allergen, the allergen binds to its antibodies, which are already bound to the mast cell, and this will now trigger this response which I just described. So this is why when we are being tested for allergy, we will the presence of antibodies against different allergens will be monitored. But this is just one way to activate the mast cells. There is also a large family of secretagogues or stimuli called basic secretagogues because they are all positively charged, and they can also activate mast cells, leading to the same responses which I just mentioned. We know about them that they activate G proteins, which I will come back to in a few minutes, and by this they can activate a cell, and like the IgE allergen, will again induce the activation of the cell and the formation of and release of inflammatory mediators. And another way to activate mast cell is through cell-cell contacts. These cells can contact other cells like eosinophils, T cells, nerve cells, and many others. And through this cell-cell contact, they undergo activation and again result in the same inflammatory process. So we have both IgE-dependent as well as IgE-independent allergic reactions, as well as inflammatory reactions which follow the immediate allergic reaction. And therefore, it is not surprising that we find the mast cells being involved in many, many inflammatory reactions. The classical allergy, which is IgE-dependent, also referred to as atopic allergies, but also many other sorts of allergy, like neurogenic inflammation, because this family of basic secretagogues, these positively charged molecules, many of them include neurotransmitters, which could also therefore activate mast cells. So not only an allergen which will induce an immunological reaction will activate the mast cell, but sometimes change in temperature that will trigger release of a neurotransmitter will then trigger activation of mast cells, leading to allergy in response to a change in temperature. Also stress, some changes in our mood may affect release of neurotransmitters and then subsequently leading to activation of mast cells, and allergy, which is in fact based on change in our mood or exposure to stress conditions. In addition, mast cells also have a very significant amplifying role in many other inflammatory diseases, such as autoimmune diseases, inflammatory chronic diseases like asthma, tumor growth and angiogenesis, and many other processes in which mast cells become activated through each of these mechanisms, any one of these mechanisms leading to the activation and release of these mediators. I already alluded to the fact that current therapies suffer from disadvantages, and in fact there isn't a real treatment for allergy, and we only deal with the symptoms of this disease. And if we go and we think about them, if we look at the therapies that are available, then obviously they include antihistamines, which obviously if we understand the number of inflammatory mediators that mast cells can produce and release, then obviously antihistamine could not counteract all the inflammatory reactions. It could neutralize the effect of histamine, but for sure all the other inflammatory mediators will not be affected by an antagonist to histamine. Same goes for anti-leukotrienes, another drug that is available. It could counteract the effect of leukotrienes, but it will have no effect on the biological activity of histamine or cytokines or chemokines or all the other mediators. So for sure, this kind of treatment is restricted and limited in its potency. Steroids are often used to treat allergic reactions. They are effective, but even this family of drugs, which is effective, is not effective to a subset of mediators. And Ronit has a poster showing that some of the very important mediators that are released by mast cells and play a very important role in the pathology of asthma are in fact not affected by steroid. In fact, they are even enhanced by steroid. And in addition, of course, we all know that steroid treatment is associated with severe side effects. Human anti-IgE, which is another therapy which was developed, will of course be effective only against IgE, but not against IgE-independent allergies. So there is an important and urgent need for a therapy that could deal with all the mediators that are being produced and released by mast cells. What we would like to have is a drug that could inhibit this cell so that none of these mediators will be released, and only this could be effective against the pathological outcome of activation of such cell. So if we look again at this cartoon and we think what could be an effective way, in fact, to inhibit all these mediators, then of course one way that comes into mind is if we could learn about the signaling pathways that are triggered, initiated in the cell when the cell encounters an external stimulus, and are responsible for these cellular processes. If we could find the hubs, the common signaling points that are common to all the secretagogues, that are common to all the mediators, and if we could block those ones, then we could come up with a way to treat allergy as we would prevent the activation of the cell and therefore simultaneously prevent the release of all mediators, irrespectively to the kind of stimulus that has activated the cell and to which class of mediators is being released. So this, in fact, is the challenge that we are trying to address. We are trying to understand or to identify those signaling molecules that will be crucial for the activation of the cell so that we could look at them as potential targets for drug development. So this is the signal transduction-directed drug approach, and as I already mentioned, specifically I will be discussing also cell-permeable peptides as potential tools. So how do you do it? How are you going to identify signaling molecules that play a crucial role in the activation of a cell? So in fact, this question is not so problematic, or it shouldn't be so problematic with the knowledge that we have today, with the whole genome being available to us. In principle, it shouldn't have been such a problem because we can think about a system where we would put our cells, let's say our mast cells, in a tissue culture dish, and we would then stimulate them with an external stimulus, and we would then measure histamine release, which for us will illustrate the activation of the cell. Now, if we want to identify something, a gene that is important for this process, we could introduce into the cell what I call here a modulating gene, which could be a double-stranded RNA that will block the expression of a gene, or it could be a mutated gene that will act as a dominant negative inhibitor. And we could then, in principle, ask, how does the expression of such a perturbation affect histamine release? So this is the way to go, only that with mast cell we have a problem. The problem is that the transfection yield, the yield by which we can introduce a gene or DNA into the cell is very limited. As shown in this cartoon, if we were to do it on mast cell, we would get only a few cells to take up this modulated gene and be affected and express it. Therefore, if we were to measure histamine, then the modulation of this subset of cells will have no impact on the average readout. Namely, we will mainly see the cells which are not at all transfected, and therefore, using such an approach, we will be unable to evaluate the effect of this particular gene which we have tested. So in order to overcome this problem, what we do in the lab is to try and use, instead of looking at an endogenous mediator that is being released by the cell, to introduce into the cell a reporter. And Ronit has a poster which describes in detail this approach that we are taking in the lab. So what do we mean by reporter? We want to introduce into the cell a gene whose product will behave exactly similar to the endogenous mediator, so that it will go to the same granules and be released under the same conditions that the endogenous mediator is being released. Moreover, we will introduce it into our cells. The system will be calibrated in a way that the reporter will always go inside together with our modulating gene. So each single cell that will express the reporter gene will also express our modulating gene. And then instead of measuring histamine, which comes from all the cells, if we only measure the reporter, then we are sure that we are in fact measuring only the population that is also expressing our modulating gene, and therefore we can really evaluate the real impact of the modulating gene on the activation process. So to do this, what Ronit has done was to choose a reporter we are using a peptide, neuropeptide Y, NPY, which is conjugated to a fluorescent protein, which makes it easily detectable. And of course, what we have done was to make sure that indeed this reporter is expressed and released exactly as the endogenous mediators. So here we just see a picture showing serotonin is an endogenous mediator like histamine, and it's found in these granules. And we see that when we express in the cell our reporter, NPY, RFP, red fluorescent protein, they have the same pattern and they co-localize. Moreover, under conditions that the endogenous mediators are being released, when we try different external stimuli, or if we try different concentrations of a stimulus, always the endogenous mediators are being released similarly to the reporter, and all the details are in the poster. So with such system, we can now monitor only the reporter in cells which are genetically manipulated and assess the impact of our manipulating gene on this process. To be sure that indeed this is what we are doing, in collaboration with Nomit Surgil from the Department of Physics and the Institute of Interdisciplinary Biophysics and Technology, Nurit is using cell-retaining chip array, which really traps single cells individually in wells. And this way, we can monitor each cell to be sure that indeed it does co-express our modulating gene or a control gene. And we can monitor the release process in living cells, monitoring the kinetics, monitoring correlation with the expression level of the manipulating gene, and many other parameters that can be changed and followed. Moreover, by zooming into the cell, one can also correlate the impact of our modulating gene on our release process by looking at the cellular localization of the monitoring of the manipulating or modulating gene, so as to see at a correlation between the cellular localization of a protein and the way it can affect the secretory process. So these chips can be coated by different materials, and this is one point which I wanted to raise. As I said, I hope also to come home with some future collaborations. These chips can be coated by different polymers and therefore allow to learn the interactions between coating of this chip and the responsiveness of the cell. So again, I won't go into detail because the poster is outside. Nurit has used this technology in order to ask how do RAP proteins, which is a family of GTP binding proteins which are known to affect transport in a cell. So she has screened the whole family of the RAP GTPases to ask how do they affect mast cell activation, mast cell secretion. And she has monitored each one of these cells to see whether it is expressed in the cell and where exactly is it localized in the cell. And using this technology, she was able to cluster these GTPases and see which ones are mostly involved in this response, so that if we introduce a dominant negative mutant, they will inhibit the secretory process, as opposed to others whose inhibition rather potentiates this response. So this is an approach where one can identify signaling molecules in mast cells. Now, having identified such molecules, how do you proceed? How do you place them at a position of drug targets? Can you really use this information in order now to inhibit them? So here I would like to give one approach that we have been adopting and trying, and this is based on the formation of cell permeable peptides, which are specifically directed against such target. And the target I would like to describe is a G protein called GI3, which we have identified as playing a role in mast cells. So just briefly, what are G proteins? G proteins are a family of proteins, and they are assistants for receptors and enable them to promote the cellular responses. So we have a large family of G proteins which couple to even a larger family of G protein coupled receptors, and they can mediate many important cellular responses, metabolic responses, immunological responses, synaptic transmission, and so on in our body. So what's important for us to understand is that, in fact, a G protein is a protein that binds GDP. It is found in close proximity to its receptor, which is a G protein coupled receptor. However, when a ligand binds to a receptor, then this protein becomes active by exchanging GDP for GDP and then producing a cellular process. Now, it was shown these G proteins consist of 3 subunits, alpha, beta, gamma, and it was shown that peptides that comprise part of the alpha subunit can compete with the G protein and inhibit the interaction between the receptor and the G protein, and therefore also inhibit cellular responses. So we have used this information, namely the ability of peptides to compete with their own G protein, in order to identify G proteins that may play a role in mast cell activation. And you can see here 2 peptides. One corresponds to a G protein called GI2, and another corresponding to GI3, and only the peptide which corresponds to GI3 is able to inhibit histamine release when introduced into permeabilized cells. Now, can we use this information in order to have a peptide that could also have a therapeutic potential? For this, of course, we have to overcome the permeability barrier. Peptides do not penetrate cells. So we have produced a chimeric version of such peptide, of the GI3 peptide, where the functional domain was conjugated to a leader domain, which serves as an importation sequence and allows this whole peptide now to penetrate into cells. We could show that such a peptide could indeed inhibit histamine secretion also from intact cells, so we did not need to permeabilize the cells. This peptide could penetrate the cells and inhibit the response. This slide, all these black lines, just illustrate activation of a cell. When a cell undergoes activation, proteins are being changed, and this is illustrated here by this signal that appeared when the cell was activated and is absent here in cells which are not activated. And what you can see here, that this peptide, when it is introduced into the cells, it inhibits the whole signaling pathway. So we have a peptide that does indeed inhibit activation of mast cells. Does it have any therapeutic potential? The answer is yes, because when we tested it in vivo on skin allergy or in an animal model for asthma, we can see that in both cases this peptide, which we termed IL-1, could inhibit the skin allergy which was induced by an allergen, or in the asthma model, when we measured airway resistance, which increases when asthma is induced by an allergen, we can see here that the peptide could inhibit this response. So basically, we believe that such an approach, where we look at, for example, at a signaling molecule, could really be a useful tool in order to inhibit signaling pathways, and also be of potential future therapeutic. Again, as I said, I'm not going into the molecular mechanism of this process because we have a poster and Ronit will be happy to describe it. I just want to mention one thing which might be relevant to this audience, and I hope to interest the audience in this point. We noticed that this peptide, which consists of 16 amino acids, which are the hydrophobic domain, which is the importation sequence, and 10 amino acids, which are the functional inhibitory domain, where this is a alpha helix, and here is a proline which combines the two parts of this peptide. As you can see, it is 26 amino acids. The molecular weight should be around 2,600. But if you run it on a gel to estimate the molecular weight, the major band you get is of 10,000. So if you look at this peptide, you see that it self-assembles to produce larger structures. In fact, we can, if we look for longer exposure time, we can see that even higher molecular weight structures are being generated. So since we know that this is a alpha helix and this is a proline, it looks as if we have here an assembled structure which consists of 4 peptides giving this tetramer. And this, of course, raises the questions like, are there any restrictions to what size of this peptide could self-assemble? Does it form any regular or any structure when it self-assembles? Does it form a nanotubules, or is it just a non-structure randomal conformation? Who is the functional entity? Is it this tetramer, or is it the monomer, which is in fact responsible for the functional activity? So these are questions that I hope that maybe we could initiate some collaborations. So in summary, I've shown that a reporter-based functional genomic analysis enables the identification of key signaling proteins in mast cells, that cell-permeable signaling-based peptides are powerful tools for dissecting mast cell activating pathways, and finally, that such peptides do also display some potential therapeutics and may serve as base or as future therapeutic tools for allergy treatment. So I already mentioned that Nurit, who is here and has a poster, is carrying out the functional assay in order to identify signaling molecules in mast cells, together with Nomit Sorgil from Bar Ilan, and Matsadaw is helping us with the bioinformatics, and Fukuda, who is providing some of the constructs. And the GI3 work was initiated with Tamiraz, Orly Zavar, and Irit Scheffler, and continued by Dana Baram and Ronit Dekel, who is here in the audience and has a poster on it. And I thank you very much for your attention.
Thank you, Ronit, for a very comprehensive introduction. It was great. Any questions? Yes, Ron.
Are these mast cells also good for anything?
Well, this is a very good question, and the answer is that in fact, yes. They do have some physiological, it was shown first that they are immune cells. They are important also against bacteria, so that they are innate immunity. Also in wound healing, they play a role. So this is another reason for the importance of studying their signaling pathways, because instead of just blocking them or eliminating them by inducing, for example, apoptosis, it would be much better to identify all their signaling pathways, cluster them with respect to cellular processes, and then to try and find targets that will not hamper the physiological functions, but only the pathological ones.
I think we should add one important mechanism to complement activation.
You are absolutely right. It falls into the basic secretor box. The complement generates peptides which are positively charged, and they do activate the cells IgE independently under the family of basic secretor box.
There are a few types of activation, but that's not
yeah, we can speak now about the activation of the muscle itself.
Yeah.
You are right.
Okay, so thank you for both.
Fine. Okay, thank you.